Welcome, students. Am I audible?
Good afternoon, sir.
Yes, sir.
Yes, sir. Good
किसका
afternoon.
presentation है आज?
Good
Presentation
afternoon,
बताया था?
sir. Yes, sir.
Good afternoon. किसका
Good afternoon,
है?
sir.
Nandini.
Yes, sir.
So, are you ready, Nandini?
Yes, sir.
Okay, then share your slides. And also, while presenting, turn on your camera. और नंदिनी के बाद किसका है? Who is after Nandini?
So, Saiba, me.
Okay, Saiba. So both of you be ready for that. And so first,
Okay,
Nandini
sir.
is going to present. I think Nandini will present on DNA, and Saiba is going to present on RNA. Am I right?
Yes, sir.
Okay, so start.
Sir, is it visible?
Yes, slides are visible.
Okay, all right, sir. I'll start then. Good afternoon, everybody. And today I'll be talking about DNA and the structure of DNA. So basically, first I'll give a brief introduction to DNA. So DNA, or the deoxyribonucleic acid, is the building block of all the human body. It carries the genetic material of all the living beings as well as certain viruses. They code genetic information within them and are found in all eukaryotic organisms as well as prokaryotes. The shape and size may differ in each type of cell. It's a nucleic acid, and it is one of the four major types of macromolecules that are known to be essential for all.
Nandini, also turn on your camera. Nandini, turn on your camera. Nandini, you got muted also.
All right, sir. So it says you can share your video only after you're done screen sharing. So I think that's why I'm not visible.
Okay, continue then, Nandini. Continue in the same way.
Okay, sir. And DNA is found in the nucleus with a small amount of DNA also present in the mitochondria in the eukaryotes. Next, I'll be talking about who gave the first model of our DNA. The chemical DNA was first discovered in 1869, but its role in genetic inheritance was not demonstrated until 1943. In 1953, James Watson and Francis Crick, aided by the work of biophysicist Rosalind Franklin and Maurice Wilkins, determined that the structure of DNA is a double helix polymer, a spiral consisting of two DNA strands wound around each other. This is the most probable timeline that has been accepted for the discovery of DNA, but but only a few people know that approximately 50 years prior, Swiss physiological chemist Friedrich Miescher first identified what he called the nuclein inside the nuclei of human white blood cells. The term nuclein was later changed to nucleic acid and eventually to deoxyribonucleic acid, or the DNA. After that, another Russian biochemist, Phoebus Levin, worked on it, followed by Erwin Chagraf, who was one of the handful of scientists who expanded on Levin's work by uncovering additional details on the structure of DNA, thus further paving the way to Watson and Crick. Only then we got the standard double helix model in the final form. Next, I'll be talking about the features of the Watson-Crick model. So the first feature that was proposed was that DNA is a double-stranded helix with two strands connected by hydrogen bonds. A bases, or the adenine bases, are always paired with T bases, and C bases are always paired with G bases, which is consistent with and accounts of Chagraf's rule. Secondly, most DNA double helices are right-handed. That is, if you were to hold out your right hand out, with your thumb pointed up and your fingers curled around your thumb, Your thumb would represent the axis of the helix and your fingers would represent the sugar phosphate backbone. Only one type of DNA, called the Z-DNA, is left-handed. The third rule was that DNA double helix is antiparallel, which means that the 5 end of one strand is paired with the 3 end of its complementary strand, and vice versa. This I'll be showing in the next figure. And nucleotides are linked to each other by their phosphate groups, which binds the 3 end of one sugar to the 5 end of the next sugar. The last point is that not only the DNA base pairs are connected via hydrogen bonding, but the outer edges of the nitrogen-containing bases are exposed and available for potential hydrogen bonding as well. These hydrogen bonds provide easy access to the DNA for the other molecules, including the proteins that will play a vital role in the replication and expression of DNA. This is a diagrammatic representation of the double helix model that was proposed by Watson and Crick. So as you can see, there are two hydrogen bonds which connect the T base and the A base, and there are three hydrogen bonds which are connecting the G base to the C base. The sugar phosphate backbones, which are shaded in gray, run anti-parallel to each other, and the 3 end and the 5 ends are aligned to each other. So basically, what we summarized was that each strand of a DNA molecule is composed of a long chain of monomer nucleotides. The nucleotides of DNA consist of a deoxyribose sugar molecule to which it is attached a phosphate group and one of the 4 nitrogenous bases. 2 are purines, which are adenine and guanine, and 2 are pyrimidines, which are cytosine and thymine. The nucleotides are joined together by covalent bonds between the phosphate of one nucleotide and the sugar of the next, forming a phosphate-sugar backbone from which the nitrogenous bases protrude. One strand is held to another by hydrogen bonds between the bases. The sequence of this bond is specific, which means that adenine will only bond with thymine and cytosine will only bond with guanine. This is called the complementary base pairing. Now I'll be talking about the molecular structure of DNA. So this is a well-established fact that DNA molecules are basically polymers. Polymers are large molecules that are built up by repeatedly linking together smaller molecules called the monomers. Each strand has a 5 end, which is a phosphate group, and a 3 end, which is a hydroxyl group. Next, I'll be talking about the strands, which are basically anti-parallel, which means the 5 end will only be next to a 3 end, and the other one at the 3 end will be a 5 end. There is absolutely no instance or any structure of DNA where the 5 end and the 5 end are aligned to each other. These deoxyribonucleotides are linked together by a phosphodiester bond. The nitrogenous bases that compose of the deoxyribonucleotides include adenine, guanine, cytosine, and thymine. The complementary of the strands are due to the nature of nitrogenous bases. The base of adenine always interacts with a thymine on the opposite strand via 2 hydrogen bonds, and cytosine always interacts with guanine via the 3 hydrogen bonds on the opposite strand. The shape of the helix is stabilized by hydrogen bonding and a hydrophobic interaction between the bases. The diameter of the double helix is around 2 nanometers, and the double helical structure repeats at an interval of 3.5, which corresponds to 10 base pairs. Next, I'll be talking about the structural diversity in DNA. So DNA, the double helical structure that I explained before was called the primary structure. There are other 2 different types of structures. The previous one was more of a 1D structure. Now I'll be talking about the secondary structure and the tertiary structure in the following slides. So the sequence or the order of the nucleotides define the primary structure of the DNA and the RNA. The secondary structure, which I will talk about now, is a set of interactions between bases, which means the parts of strands are bound to each other. In a DNA double helix, the 2 strands of DNA are held together by hydrogen bonds. The secondary structure is responsible for the shape of that nucleic acid, which are bound to each other. The base in the DNA are classified as purines and pyrimidines. DNA's secondary structure is predominantly determined by base pairing of the two polynucleotide strands wrapped around each other to form a double helix. Although the two strands are aligned by hydrogen bonds in base pairs, the stronger forces holding the two strands are stacking interaction between the bases. These stacking interactions are stabilized by van der Waals forces and hydrophobic interactions and show a large number of local structure variability. There are also two grooves in the double helix, which are called the major groove and the minor groove based on their relative size. Over here, as you can see, I am trying to show that in the double helix structure, when the sugar phosphate backbone is not equal all over. This is due to the disparity in the length of the hydrogen bonds between the nucleotides. This is where the difference in the difference between nucleotide bases gives rise to another type of structural diversity, which I'll be talking about now, which are the minor and major grooves of the DNA. So as mentioned earlier, a result of double helical nature of DNA, the molecule has two asymmetrical grooves. One groove is smaller than the other. This asymmetry is a result of the geometrical configuration of the bonds between the phosphate, sugar, and the base groups that forces the base groups to attach at a 120-degree angle instead of a 180-degree angle. The larger groove is called the major groove, which occurs when the backbones are far apart, while the smaller one is called the minor groove, occurs when they are close together. Since the major groove and the minor groove expose the edges of the bases, these grooves can be used to tell the base sequence of a specific DNA molecule. The possibility for such recognition is critical, since proteins must be able to recognize specific DNA sequence on which to bind in order for the proper function of the body and cells to be carried out. Now I'll be talking about the tertiary structure of DNA. Tertiary structure refers to the location of the atoms in a three-dimensional space, taking into consideration geometrical and static constraints. It is a higher order than the secondary structure, in which large-scale folding in a linear polymer occurs and the entire chain can be folded into in a specific three-dimensional shape. The tertiary arrangement of DNA's double helix in space includes bDNA, aDNA, and zDNA. Triple-stranded DNA structure have been demonstrated in repeat polypurine or a polypyrimidine microsatellite sequences and satellite DNA. bDNA is the most common form of DNA in vivo and is a more narrow, elongated helix than aDNA. It has a wide major groove and it makes up which is more accessible to proteins. On the other hand, it has a pretty narrow minor groove, and bDNA's favored conformations occur at high water concentrations. Next is the aDNA or the A form of DNA, in which the DNA duplex which is observed under dehydrating conditions. It is shorter and wider than bDNA. aDNA has a deep, narrow major groove, which does not make it easily accessible to proteins. On the other hand, it has a very wide, shallow minor groove, which makes it accessible to proteins, but at a lower information content than the major groove. Its favored conformation is at low water concentration. Lastly is the zDNA, which is a relatively rare left-handed double helix. Given the proper sequence and superhelical tension, it can be formed in vivo, but its function is unclear. It has a more narrow, more elongated helix than A or B. zDNA, major DNA, is not really a groove, but it has a narrow minor groove. The most favored conformation of zDNA occurs when there are high salt concentrations. So basically, in the tertiary structure, there are four types of DNA. The aDNA, which was originally identified by X-ray diffraction of analysis of DNA fibers, and it has a 75% rate of relative humidity. Next is the bDNA, which is the most common type of DNA found, and it was also originally deduced from X-ray diffraction of sodium salt of DNA fibers, and it has a high relative humidity content, around 92%. Third is the cDNA, which is extremely rare and is formed at 66% relative humidity and only in the presence of lithium and magnesium ions. Fourth, we have the zDNA, which is the only left-handed double helical structure, and it winds more to a zigzag pattern. Next, I have the diagrammatic representation of each types of DNA. As you can see, the aDNA, the minor groove and the major groove are a bit different and are more closely packed. bDNA has a more standard structure that we know, that is most commonly represented. cDNA has the helical structure is a bit more far apart. The backbones are a bit more far apart than the other two, and zDNA has a more irregular zigzag type of pattern to it. Lastly, I'll be talking about the effects of structure on the function of DNA. So first we have the replication. The structural features that allow DNA to replicate or copy itself perfectly are its double helix. I'm sorry, did somebody say anything?
No, Namini, continue. Might be someone turn on the mic in between.
Okay, sir.
Okay,
All
so
right,
continue.
sir. All right. Sorry. Structural features that allow DNA to replicate or copy itself perfectly are its double helix and the true standard shape made up of complementary bases. Next, making of proteins. The DNA carries the genetic information for making proteins. The base sequence determines amino acid sequence in proteins. Messenger RNA or the mRNA is a molecule which carries a copy of the code from the DNA in the nucleus to a ribosome, where the protein is assembled from the amino acids. Lastly is the evolution. An organism's DNA affects how it works, how it behaves, and its physiology. So a change in an organism's DNA structure can cause changes in all aspects of its life. Mutations are essential to evolution. They are the raw material of genetic variation. Without mutation, evolution could not occur. In a more simpler way, the gene or the sequence of DNA ultimately determines the unique sequence of amino acids in each peptide chain. A change in nucleotide sequence of the gene's coding region may lead to a different amino acid being added to the growing peptide chain, causing a change in protein structure and therefore function. Thank you for listening.
Nice presentation, Nandini.
Thank you, sir.
But you covered lots of very important things in your presentation. Mainly, I would like to refer about the groups. And but lots of things are missing in your presentations also. One thing is the way that you present it just simply look like that you are reading the text, just looking over the slides and whatever the texts are there on your slides, you are just reading that. Okay, so and you're not explaining anything, so there is no explanation, just you're reading, keep on reading, continuously reading. Look like that you somewhere you from the web you got a presentation and you presented it as it is. And second is the missing images were there, so you simply just referring reading the text and just showing on a that this is the image and how these are going to be to what you're referring. Like you simply mentioned that what's in quick structure, then what is the pitch is going to be, what is the height, so how you're referring that, what you're it's completely missing in your presentation. And second thing, one more thing that is missing in your presentation is you didn't tell anything about the nucleic acid. You didn't mention anything about the sugar, you didn't mention anything about the bases and how they are connecting, how they are linked. Nothing was there, simply just only you're explaining the Watson-Crick model. When I mentioned at your presentation about the DNA, it means you have to cover all the aspect of that. That means you have to talk about you're starting with your nucleic acid, it's more nucleotides, nucleoside. So how this sugar and phosphate is attached, how the sugar and nitrogen base is attached, at what position they are attached, what is the numbering, what is the sugar in, why you are calling it deoxyribose sugar. So what sugar is there? You didn't mention anything about it. So lots of things are missing in your presentation, but one thing is that really some information were quite good and really Student speaks about that. You bring those information here. It's a really nice thing, and even we also rarely bother to give you the details of those features that you mentioned in your presentation. But the one thing missing is that you didn't explain those things. You didn't make us to understand what you want to say. Just reading the text doesn't help. It is also not going to help you also, and is not also going to help your listeners also. Okay, so before moving to Saiba, now we have to move to our listeners. So, listeners, do you have any query from Nandini? Listeners, presentation open for discussion. Yes, Nehal, please ask.
Sir, am I audible?
Yes, Nehal, you are audible, but speak a bit loudly. Your voice is very feeble,
Yes, sir.
and you can turn on your camera also if you are in a position to turn it on.
Yes. Sir, am I visible?
Not visual, and your voice is very faint. Please speak a bit loudly or come. Yes, you are visible.
Yeah. So, sir, my question for Nandini is, Nandini, what is the basic function of the minor grooves in the DNA?
So basically, the binary groove of DNA it provides a unique chemical environment to the proteins because around the minor groove there is the polarity. There is a lot of polarity, and the electronegative potential is very high. And the walls are lined with H bonds, but still there are no H bond donors. So basically, the minor groove is it basically steers the proteins toward the major groove, and it helps in in DNA in recognizing the correct DNA sequences.
Okay, thank you, Nandini.
Nihal, first thing I would like to ask you, do you got the point what really these grooves mean? Because asking the question first, we should understand what the groove mean. Okay, so do you got Nihal what really this groove mean? Whom we are calling the grooves? Because it's not just in a two-dimension figure. It is really very difficult to understand whom we are referring the grooves. Just by showing that this image that some later type of thing is there, one part we are putting keeping this is minor and that is major. So for understanding this groove business, we should listen in a three-dimensional way. And when we're referring the three-dimensional way, Nandini, please show that circle image that you shown na? The circle one part
It's
you
a
showed in your slide. Please show that slide.
it's a this one, sir.
Yeah, this one. Yes, this one. Yeah, so please make it big.
It's a
Okay, so here you can seeing that upper one, the circle is there. So in this circle means you have to see in this way, like you're audible to me. It's like this is your, like if you can see, this is the staircase we are mentioning, and the staircase is going like this, but we have to see it like that. And when you see your staircase like that, you will found that the division, the symmetry is not there. Means your nucleotides are more in one part and lesser in the another part. So that simply you can say draw a circle and cut it like in this way, like this is a thing. This is notchy type of things are going to be there. When this notchy type of things is there, in it you can see this is a lesser space in this in this my between of you can say my fingers, and then more at this angle. So like this side, this is a more space. So this we are telling these are the grooves. This side where the more angular things are there, that is the major groove, and this side is going to be a minor groove. So that you can when you can see here also, if you as you can see one line is there that she drawn a line, and this line is telling you that distribution is not equal. So if you see the left side of the line, you are going to have a more thing compared to the right side of the line. So this the area where the more components are there and notchy appearance is going to be there. It means more nucleotides is there. To that we are calling it as a major groove. Wherever on the left right side where the less number of space is there, so that we are calling it as a minor groove. So this it is a simply it is a three-dimensional things. It is very difficult to understand in two dimension. So in our next class I will show you, I will try to explain you, means in your when I will talking about, I will also give you some idea of that. At that time I will explain you much, I will try to explain you in detail. I'll also show you some videos also, so that with this video will explain you in better way. Okay. And the things as Nandini was mentioning you, the role of these major and minor groove. In principle, these major and minor groove, as Nandini mentioned itself, they are the intermolecular interaction, and that interaction leads because of the hydrogen bond between the two nucleotides. Okay, so when they are packaging is going to be, it's not a straight, it is a helical type of the arrangement. So secondary structure that you are referring. That's why. Okay, so in that thing is there, so because of that interaction it is going to be like that, but that interaction further leads to for more interaction. So major groove going to have more surface exposed, so that you can say that it is going to be exposed more molecules which are non-DNA in nature, like it can expose to some proteins also while it is forming the structure of your chromosomes. And minor groove, it's being going to be very lesser space, less volume in it, so it also help in the capturing, so like small molecules which can penetrate inside it, so that it can capture it. Like simply if you remember, when we are talking about your detection like electrophoresis, if you remember, and we mentioned the etherium bromide is going to be there. Etherium bromide go and intercalate in the minor grooves. So minor grooves means the smaller the pouches. This pouch is there that so here ethylene bromide go and stuck here, it got packaged here, simply you can say. However, it is not it's in this interaction is going to be useful for us, but this interaction is not no way beneficial for a DNA. But some other type of the interaction definitely going to be there that help in the opening up of the DNA. Like you want to open it up, like one thing, supercoiling is there. So to if you have to change your supercoiling, so these grooves help in providing the site for the action of various enzymes also. But uh, this is the result of those grooves, but DNA do not make these grooves for that purpose. These grooves are there because of the intermolecular interaction within your DNA. Okay, hope Nehal, I explained your question. So now moving to next,
Yes, sir.
who is going to ask next question? Next query. No one. Okay, so we can move to Sahiba. Sahiba. Yes, Namrita want to ask. Yes, Namrita, please turn on your mic and ask.
Nandini, what does groove do?
Namrita, I think you wake up late. This whole from that the Nehal asked the same question, and since then I'm answering this question itself, and you asked again the similar same thing. Were you here itself or Namrita अभी ज्वाइन किया है
No,
नम्रता?
sir.
तो फिर जो मैंने इतना लंबा चौड़ा बोला सुना था वो? हम्म?
Yes, sir. Yes, sir.
Then why you ask the same thing again? Okay, so Nandini, please answer it again. So Namrita wake up just now. So please answer it again.
So basically, the groove, what the groove does is helps in the DNA sequences and the protein. So the minor one was steering the protein, and the major one is it's important for the DNA binding proteins, which is involved in the replication and transcription. So basically, they recognize specific DNA sequences in like the shortest way.
Okay, so who are going to ask next? So Nehal, Amrita, then who will be the next? It's really nicely Nandini presented the thing. I hope many of you would like to ask questions. No one? Okay, so thank you, Nandini, for your nice presentation. But Nandini, keep those points as I mentioned you.
Yes, sir.
So while making presentation, don't just download it from the Google and came up with that. Okay, you have, at least you don't want to do more on the presentation you got. At least study it thoroughly so that you can explain. So the
Okay, sir.
explanation must be there. That was completely missing in your presentation. Okay,
Okay,
so
sir.
please
I'll keep
follow
it in
these
mind.
things. Okay, so hope Sahiba is also going to share the similar things, but she's going to talk about the RNA. So hope she will bring these points that I mentioned to you. Okay, Sahiba, please share your content and also turn on your camera while presenting. And like if you share your screen from your desktop, might be the camera got off, or if you are working from the your web directly, so that at that time you can't turn on your camera. But if you are downloaded the app and you're opening through app, then camera you can turn on the camera. Okay, so these are the issues while you are presenting, whether you're presenting using your browser or using your app. Okay, so Saiba, please continue.
Okay, sir. So Sam will be uploading my slides, so I will turn my camera on.
Okay, so Demetrius. Yes, Saiba, start.
So, am I visible?
Not yet, Saiba.
So I've turned my camera on. So I'll start.
Yes, you can start.
Okay. So, good afternoon. Today I will be giving a presentation on the topic structure of RNA. So, first I would like to start with the basic introduction. So, ribonucleic acid RNA is a polymeric molecule which is essential in various biological roles like coding, decoding, regulation, and expression of genes. Along with lipids, proteins, and carbohydrates, nucleic acid constitutes one of the four major macromolecules essential for all known forms of life. Like DNA, RNA is assembled as a chain of nucleotides, but unlike DNA, RNA is found in nature as a single strand folded onto itself rather than a paired double strand. RNA is a polymer of ribonucleotides linked together by phosphodiester linkage. Cellular organisms use messenger RNA, known as mRNA, to convey genetic information using the nitrogenous bases of guanine, uracil, adenine, and cytosine, denoted by the letters G, U, A, and C. That directs synthesis of specific protein. Many viruses encode their genetic information using an RNA genome. Some RNA molecules play an active role within cells by catalyzing biological reactions, controlling gene expression, or sensing and communicating responses to cellular signals. One of these active processes in protein synthesis, uh, a universal function in which RNA molecules direct the synthesis of proteins on ribosomes. This process uses transfer RNA, tRNA molecules to deliver amino acids to the ribosome, where ribosomal RNA, rRNA, then links amino acid together to form coded proteins. Now, talking about the basic history, like the history of RNA. So it was the first genetic material, and in 1967, Carl Woese found the catalytic properties of RNA and speculated that the earliest forms of life relied on RNA both to carry genetic information and to catalyze biochemical reactions. Their theories were not validated until the work of Nobel uh, Prize laureate Thomas R. Koch. In the 1970s, Sech was studying the splicing of RNA in a single-celled organism when he discovered that with an unprocessed RNA, the molecule could splice itself. He announced his discovery in 1982 and became the first to show the RNA has catalytic functions. Moving on to the next slide. Now I will talk about the structure. So there are three main components. The first one is phosphate group. The second one is sugar, ribose, and nitrogenous base. The nitrogenous base are divided into two groups, purine and pyrimidine. Purines is made of six-member ring fused to a five-member ring, adenine, guanine, and pyrimidine is made up of six-member ring, cytosine, and uracil. Then the nucleotides are formed by the condensation of a sugar phosphate and one of the four bases. The following illustration represents one nucleotide. The base plus sugar is equal to nucleoside. Uh, for example, adenine plus ribose is equal to um, adenosine. And base plus sugar plus phosphate is equal to nucleotide. For example, adenosine monophosphate, adenosine diphosphate, and adenosine triphosphate. Then there is covalent bonding between the nucleotides. Nucleotides are linked together by covalent bonds called uh, phosphodiester linkage. Here is a structure. Then coming to hydrogen bonding, usually RNA is single-stranded, but in some viruses, RNA is present in double-stranded form. The bases in two strands are paired through hydrogen bond, H bonds, forming base pairs. Adenine forms two hydrogen bond with uracil from opposite strands and vice versa. Similarly, guanine is bonded with cytosine with three H bonds. And here is talking about the double-stranded DNA structure. So there are double-stranded DNA structures. DNA can fold back on itself. It depends on the base sequence, and it gives stem, double-strand, and loop single-strand structures. Double-stranded RNA has an A-like conformation, and steric clashes between two OH groups prevent the B-like conformation. Has a structure. Now, to summarize the structure of RNA, each nucleotide in RNA contains a ribose sugar with carbons numbered 1 through 5. A base is attached to the to the first position. In general, adenine, cytosine, guanine, or uracil. Adenine and guanines are purines. Cytosine and uracil are pyrimidines. A phosphate group is attached to the third position of the ribose and the fifth position of the next. The phosphate groups have a negative charge each, making RNA a charged molecule, polyanion. The bases form hydrogen bonds between cytosine and guanine, between adenine and uracil, and between guanine and uracil. However, other interactions are possible, such as a group of adenine bases binding to each other in a bulge or a GNRA tetraloop that has a guanine adenine base pairs. Now moving on to the types of RNA. So in all prokaryotic and eukaryotic organisms, there are three main classes of RNA molecules that exist. and they are messenger RNA, transfer RNA, and ribosomal RNA. And then there are other small nuclear RNA, micro RNA, small interfering RNA, and a heterogeneous nuclear RNA. I'm talking about the first type, which is messenger RNA, mRNA. So all members of the class function as as messengers, carrying the information in a gene to the protein synthesizing machinery. So the structure is that the fifth terminal end is capped with 7-methyl guanosine triphosphate cap. The cap is involved in the recognition of mRNA by translating machinery. It stabilizes mRNA by protecting it from 5 exonucleases. The three end of the most mRNA have a polymer of adenylate residues. The tail prevents the attack by 3 exonucleases. On both fifth and the third end, there are non-coding sequences which are not translated. The intervening region between non-coding sequence present between fifth and the third end is called the coding region. The region encodes for the synthesis of a protein. Here's a structure of the same. Now going to the next type, which is transfer RNA, called tRNA. So tRNA are the smallest of the three major species of RNA molecules. They have 74 to 95 nucleotide residues. They transfer the amino acids from cytoplasm to the protein synthesizing machinery. Hence, their name is transfer RNA. They are also called adapter molecules since they act as adapters for the translation of the sequence of nucleotides of the mRNA into specific amino acids. There are at least 20 species of tRNA, one corresponding to each of the 20 amino acids required for protein synthesis. And tRNA is the only RNA species that contains the nucleoside thymide. Sorry. Now there are talking about the structures. So the first one is primary structure. The nucleotide sequence of all the tRNA molecules allows extensive intra-strand complementarily, sorry, complementarity that generates a secondary structure. And then talking about the secondary structure, each single tRNA shows extensive internal base pairing and acquires a clover leaf-like structure. The structure is stabilized by hydrogen bonding between the bases and is a consistent feature. Secondary structure, talking about the clover leaf structure, all tRNA contain 5 main arms or loops, which are acceptor arm, and then anticodon arm, dihydrouracil arm, and thymide, pseudoridine, cytosine, and it has an extra arm also. Now, third one is tertiary structure of tRNA. So the L-shaped tertiary structure is formed by further folding of the clover leaf due to hydrogen bonds between T and D arm. The base paired double helical stems get arranged into 2 double helical columns, continuous and perpendicular to one another. Now, talking about the last one, which is ribosomal RNA, rRNA. Ribosomal nucleic acid, rRNA, is the RNA component of the ribosome and is essential for protein synthesis in all living organisms. The function of the ribosomal RNA molecule in the ribosomal particle are not fully understood, but they are necessary for ribosomal assembly and seem to play key roles in the binding of binding of mRNA to ribosomes and its translation. Studies suggest recent studies suggest that an rRNA component performs the peptidyl transferase activity and thus is an enzyme. It constitutes the predominant material within the ribosome, which is approximately 60% rRNA and 40% protein by weight. Ribosomes contain two major RNAs and 50 or more proteins. The ribosomal RNAs form two subunits, the large subunit and the small subunit. The large subunit RNA acts as a ribozyme, catalyzing peptide bond formation. So these are all the types of RNAs. Uh, Talking about the major types of small RNA molecules. So small nuclear RNA, snRNA, is involved in mRNA splicing. Small nuclear RNA, snORNA, directs the modification of ribosomal RNAs, and microRNA, miRNA, and short interfering RNA, siRNA, they regulate gene expression. That's it. Thank you. I'm done, sir.
Mm, nice presentation, Saiba. Okay,
Thank
so
you, sir.
now presentation open. Now presentation open for question answer. So we'll ask first. No one willing to ask questions? Devanshi? Yes, Devanshi. Turn on your camera and ask.
Sir, am I audible?
Yes, Devanshi, you are audible. Also turn on your camera while asking question.
Sir, my camera is on.
Okay.
So, Saiba, you mentioned about the tRNA. Uh, so, it is used for transfer. Okay. So, if my question is, sometimes mutation takes place in organisms' genome. So, if the mutation is happened in tRNA synthetase, which is a ligase tRNA molecule, so what will be
Devanshi, you got muted. I think you are not audible now.
Sir, am I audible?
Yes, now you are audible.
So, Saiba, my question was, sometimes mutation takes place in any organism's genome. So, what will happen if the mutation is happened in tRNA synthetase molecule, which is malformed? So, is there any impact, immediate impact on the cell that produces this type of non-functional tRNA synthetase?
Yes, if mutation occurs on the tRNA synthetase, then amino acids would not bind to tRNA molecules, as tRNA synthetase plays a vital role in translation, but not transcription. So, tRNA synthetase is the enzyme that binds specific amino acids to corresponding tRNA molecules, and then the tRNA molecules transport the amino acids to the ribosome to create a polypeptide. So, tRNA molecules are not consumed in this process, and tRNA reserves will not be depleted if tRNA synthesis were not functional.
थोड़ा सा please explain it again. So TRA, what is the role of TRA synthetase?
The tRNA synthetase, it plays a vital role in translation, but it not in transcription. So tRNA synthetase is the enzyme that binds specific amino acids to corresponding tRNA molecules.
So Saiba, question of Devanshi might be asking that if TRA synthetase is mutated, So if tRNA synthesis is not functional, then what is going to be the impact on the cell, whether the cell will survive or die?
Uh, so, like I said, that tRNA molecules will
Yes,
not
you
be
are right.
consumed in this process.
No, I we agree what you are saying. tRNA's impact, so functioning of tRNA won't affect what you say the transcription. So its role is not in the transcription. Role of tRNA synthesis is there in case of the translation. So, but she's asking if tRNA synthesis is mutated, then will you just give a speculation on that whether the cell will survive or not? As you mentioned yourself, that tRNA synthesis is essential gene, and if tRNA synthesis is not there, then that tRNA will not be synthesized, and that tRNA will not be synthesized, then simply means that amino acid will not be incorporated into the protein. And if
Yes,
that
sir.
amino acid is not incorporated in the protein, then cell won't survive. Okay, so these are the essential
Yes,
gene. If cell is not going to have a multiple copy of those genes, then cell won't survive without that gene. Okay, so they are the essential gene. Okay, Divanshi.
yes, sir.
Okay, so
Yes, sir.
one more question was there. Sanjana was thinking, I think, want to ask. So who was the next? Who was the next? नेक्स्ट कौन पूछ रहा है? नेक्स्ट क्वेश्चन. Yes, Namrata, please ask.
Sir, it's Shivani.
Okay, Shivani.
Yes, sir. One of her slides, she mentioned about HNRNA, heterogeneous, heterogeneous nuclear RNA. Can you tell me a bit about that?
Yes, Saiba, please
Uh,
explain HMRNA.
sure. In mammalian nuclei, hnRNA is the immediate product of gene transcription. So the nuclear product in heterogeneous in size variable and is very large. 75% of hnRNA is degraded in the nucleus. Only 25% is processed to mature mRNA. Mature mRNA is formed from primary transcript by capping, tailing, splicing, and
Sahib,
base modification.
Saiba, you didn't get the question. What she asked? Yes, you want to know about the HNRNA, heteronuclear RNA. So, is that
So it
HNuclear
is
RNA? You want to say that it is,
yes.
what is that?
So
That is
it is the
mRNA.
immediate. i- Yes, yes. Sorry for interrupting. Yes,
That
sir.
I think it is a nascent RNA. That is a nascent RNA, not the HNRNA. HNRNA is something different.
The hnRNA, according to what I know, it is the immediate product of gene transcription.
Okay, so Saiba, please read it again. What HNRNA is going to be? Okay, and then come up with the answer. I don't think that you are saying is that right answer. Okay, so Shivani, so there are some non-coding RNAs are there. So non-coding RNA means that are neither mRNA, that nor tRNA, not rRNA. They are not going to correspond to them. So those are they still forms. So they have some other functions like that the RNAs are there, RNA interference. So different mechanisms are there. So they are the non-coding RNA that this hnRNA fall in the class. Okay, so they have some other function than three more specified function that we know.
Okay, sir.
Okay, so
Thank you.
anyone other than the Shivani? Okay, so now we are at the end of our session. So please, so first I'd like to call two names, Atoiba and Vivek. Atoiba and Vivek. 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 Okay, so Vivek is absent today and Atoiba is already absent,
Yes, sir.
that I know.
Yes, sir.
So Vivek, at what time you joined today?
Sir, I've been having network issues, so I was trying to join in the beginning. I think I joined in the beginning, and then it didn't work, and then I kept joining like 5-10 minute intervals, but it stuck at around 12:50, 12:45, I think.
Okay, so please all of you turn on your camera while leaving the team. So for recording the attendance. So Toiba is absent, rest all of you are present. So thank you all. Please turn on camera while leaving the team. So thank you, Saiba, for nice presentation. And both Saiba and Nandini, please share your attendance on MS Team. So that will be helpful for all of your friends. So you can upload your presentation on the your assignment section or whatever the sections are available, file section, whatever section it is available, or I will create a section for all of your presentation in assignment section. So as is a part of your assignment also. Okay. So please all of you upload your presentation there. So these presentation are for your help. So download it like in Nandini's presentation, that information about rules are there. So that will be helpful for you. Okay. So thank you all. Stay safe, stay happy. Good day.
Thank you, sir.
Welcome. So leave the team.
Thank you, sir.